Hey everybody, wanted to talk about the electron transport chain and oxidative phosphorylation. So a quick overview of cellular respiration. First you have glycolysis, which takes place in the cytosol of the cell. You end up with 2-pyruvate, which can then head into the matrix of the mitochondria to form acetyl-CoA, and then which can participate in the citric acid cycle or the Krebs cycle. During these processes, during glycolysis, formation of acetyl-CoA, and the citric acid cycle, you form these reduced coenzymes, and also right here, FADH2. All of these reduced coenzymes have been collected, and they are now going to participate in the electron transport chain. Now, if you remember, these are reduced coenzymes, so we have oxidized other molecules. You can't have reduction without oxidation. You have to have both. Oxidation is losing electrons, reduction is gaining electrons. So these reduced coenzymes basically have extra electrons, and as such, they also have extra potential energy. We are going to harness that energy in the electron transport chain. And you can see by this arrow down here, we're going to make way more ATP than you saw previously from the citric acid cycle or from glycolysis. This is a much more effective way of making ATP. So, electron transport chain. When you are moving electrons from one electron carrier to another, you're going to release energy. So you can see moving here on the x-axis, you have the reaction progress as you move forward in the electron transport chain. And the y-axis right here is the relative free energy. So when these reduced coenzymes first arrive at that initial enzyme complex, you're going to remove those electrons, and in doing so, you're going to release some energy. And every time you move electrons from one electron carrier to another, you end up releasing energy. And so you can see the relative free energy is going down as you are handing these electrons off to subsequent carriers. So energy moves down as you move, as you progress in the reaction and you move down these carriers. So here is a picture of some of those carriers. So what we're looking at right here is a section of the mitochondria. So remember, mitochondria have an inner membrane and an outer membrane. And while the the Krebs cycle took place within the matrix of the mitochondria in this inner space, the electron transport chain is going to take place. It's actually embedded in this inner membrane. You have these enzyme complexes that are embedded in the inner membrane. So here is the outer membrane right here. You have an intermembrane space in between them. And then here is the matrix from which is where the Krebs cycle took place. So here is your first enzyme complex. And you can see these reduced coenzymes are donating their electrons, giving them to this electron carrier right here. As you pass off these electrons, so these two electrons right here, 2e minus, those are your electrons. As these move from NADH plus H and they move to this CoQ right here, which is an electron carrier, you release energy. This energy is used to fuel this enzyme complex, which acts as a proton pump. And so that's why you see these two H plus right here. And remember, hydrogen is number 1 on the periodic table. This 1 means it's positively charged, so it does not have an electron. So it's basically just a proton. So that's what I mean by proton pump. This H plus, this hydrogen ion, is literally just a proton. So as you move these electrons from NADH plus H, as you move them to this electron carrier, you release energy. This energy is used to fuel a proton pump. Push protons into this intermembrane space. These two electrons will then continue to another electron carrier. So from CoQ, it's going to head over here to cytochrome C. As these electrons are passed off, it releases energy. And this enzyme complex 2, this is going to also act as a proton pump, and you see more hydrogen ions, or protons, that are being pumped into this intermembrane space. Now we're going to do the same thing again. From cytochrome C, this electron carrier is going to send its electrons to oxygen. Oxygen is the final electron carrier of the electron transport chain. So as you add these electrons, these H's, this is going to form water. So this is metabolic water that we'll refer to later on. Enzyme complex, just like the other two. This is going to harness that energy as electrons are passed from one electron carrier to another. As that energy is released, it's going to be used to fuel this enzyme complex, fuel this proton pump, and pump more protons into the intermembrane space. So, main concept so far. As you move electrons from one carrier to another, you release energy. Energy is used to fuel these enzyme complexes, which are proton pumps, and you pump protons into the intermembrane space. These are charged. So what's going on here is you're generating an electrochemical gradient. Electro because this is charged, it has a positive charge to it, and chemical because you have the ion, the hydrogen aspect of it is also forming a gradient. So you have an electrochemical gradient of hydrogen ions that's built up in this intermembrane space. By the time these electrons have been given to the final electron acceptor to make water, the electrons are done. But we're not done yet with these protons that have been pumped into the intermembrane space. So by now, you've generated a high electrochemical gradient of hydrogen ions. You have many of them present in this intermembrane space, not so many of them over here in the matrix. Here's an enzyme called ATP synthase. It is going to synthesize or create ATP. So you see over here we have ADP, that's adenosine diphosphate, and you also have inorganic phosphate groups. You can put these two together and make ATP, which is what you see right here, ATP, if you have energy. That energy is what you've been making so far. The energy in this electrochemical gradient is what you're going to use to form this bond between the phosphate and this ADP. So ATP synthase is going to allow all these protons to flow through this enzyme into the matrix. So you can see these 2, 4, 6 hydrogen ions are now on the other side. As these hydrogen ions flow down their concentration gradient, this ATP synthase is able to use that energy to form these bonds. So ADP forms a bond with phosphate, forming ATP. And so you're going to be forming many ATP this way. And this is oxidative phosphorylation. So you formed a couple of ATP in glycolysis, a couple of ATP in the Krebs cycle per molecule of glucose. And those were both through substrate level phosphorylation. This is oxidative phosphorylation. You're using this electrochemical gradient to fuel ATP synthase to form many ATP.